halfway through. So our next speaker is David Clare from Cefas, who's going to be talking about food webs, bottom trawling on benthivorous fish biomass. Okay.
Yep, morning everyone. So for my talk, I'll be asking whether bottom trawling has an indirect food web effect on benthivorous fish. And this work is being carried out as part of the Pyramids of Life project. So I don't think it will surprise anyone that bottom trawling directly reduces the biomass of bottom feeding fish or benthivorous. But then an incidental effect of bottom trawling is that you disturb the seabed, and this can affect the biomass of the benthic invertebrate prey, potentially reducing it by causing mortality due to the physical disturbance, and potentially also enhancing it due to competitive release as the trawling takes out the relatively large epifaunal organisms that the benthivorous fish tend not to eat so much. So by having this effect on the benthos, we can potentially have an indirect bottom-up food web effect on the benthivorous fish species and their biomass. So essentially what we're asking here is whether bottom trawling is potentially altering the carrying capacity at the base of the demersal food web and altering how much seafood that it can produce. And then likewise, we can also look at this in the other direction, and by removing these benthivorous predators, we're potentially having a top-down food web effect by removing predation pressure and allowing the biomass of the benthos, or at least their benthic invertebrate prey, to increase. And then that could lead to some positive effects on any ecosystem functions like decomposition or nutrient regeneration that tend to be enhanced when benthic biomass is high. So understanding these indirect and direct pathways and the balance between them could potentially provide a more holistic view of the effects of trawling and also help to guide sustainable fisheries management. So to address this question, first of all, we need some data. So for the fish samples, we have over 15,000 trawl survey samples that you can see here in blue. And for each of these samples, what we've done is filtered the community. So it only includes the benthivorous species and size classes as indicated by a recent Feeding Guild study that Murray presented the results of yesterday. And then for the benthos, we have around 11,000 samples. And here we're using the benthic grabs because this gear tends to target the smaller benthic invertebrates that benthivorous fish like to eat. And then alongside, I should also say that we're using data from 2009 onwards because this allows us to match up the biological data to fishing activity using the BMS data that's available. So at this point, we have the trawling data, we have the benthos, we have the fish, but then we also need to account for any environmental drivers that may be confounding factors and influence the relationships that we're interested in studying. So for each of the benthic, each of the sites where we sample the benthos, we also obtain these environmental variables you can see on the left-hand side of the slide here. And we need to include them in our models. So looking at the distribution of the fish samples in blue and the benthic samples in orange, you can see that they're not particularly well aligned spatially, but we need to be able to match these up. So what we did is we drew a radius of 20 kilometers around the fish samples, and then we've averaged all the data within this radius, providing that it was collected within 90 days of the focal sample, and we have at least 5 benthic grab samples to describe the ecology of the benthos within this area. And then from that, we go from this rather large data set to a relatively smaller, but still somewhat large 260 meta samples that has all of the data that we need to analyze. And then just briefly on the stats, we've used generalized additive models to look at the relationships between trawling, benthos, and fish. So basically just like a linear model, but rather than telling the model that the relationship's linear, we're just letting the data dictate what the shape of the relationship is. And then we've used a technique called causal mediation analysis to partition the total effect into the direct and indirect pathways that we've hypothesized. Okay, moving on to the results. You can see these plots here at the top. The first thing to point out is that the black line just represents the simple empirical relationship if we just fit a trend line to the data. But then the lines that are in red or green, they indicate the predicted relationships if we control and hold constant all the environmental variables that are potentially confounding the relationships we're interested in. So it's these relationships in colour that we're mainly interested in. And then starting with panels A and B, what we can see is that as we have high trawling intensity, we get a low biomass of the benthivorous fish, which of course is what we'd expect. But then we also see that we have high biomass of the benthos. So potentially quite an interesting result, but at this point of the analysis, we don't really know what's going on. This could be a direct effect or an indirect effect. Moving on to panel C, this gives an idea of what's going on from a food web perspective. And first thing to say about what it doesn't show is that there's no evidence that as the benthos biomass increases, we're supporting a higher biomass of benthivorous. We actually see the opposite. So rather than seeing a potential bottom-up effect, we seem to see a negative top-down effect where the benthivorous are depleting the biomass of the benthos of their prey. That's what the evidence seems to show. But to actually formalize this, we need to do the causal mediation analysis. And when we do that, what we find is that the effect on the fish seems to be totally direct. There's like a direct negative effect due to the harvesting, no evidence of a food web effect. But then with the fish, what we see is, the benthos, what we see is an overall positive effect. And this is largely direct. 90% of the effect is direct. And the other 10% is an indirect food web effect. And I think this relatively minor food web top-down control makes sense, considering that the benthos biomass tends to be around 2 or 3 orders of magnitude higher than the benthivore biomass. So potentially it's just not a very food-limited system. And changes to benthivorous biomass doesn't really seem to make much of a dent in the biomass of the benthos. So coming back to the diagram in the first slide, this now just represents the relationships that appear to be supported by the relationship. So the red negative downward arrow is a negative effect. The green positive arrow is a positive effect. And from this, we can draw a few sensitive conclusions. The first of them being that trawling appears to enhance benthos biomass, primarily directly, but also partially indirectly by removing the predation pressure. So this suggests potentially a positive effect on any associated ecosystem functions that are driven by benthic biomass. But then coming to the main question that we set out to answer, it seems that benthivorous fish biomass is just simply depleted directly by harvesting, and we don't see any evidence of a bottom-up food web effect, which potentially makes things a bit simpler when it comes to modelling and managing these benthivorous fish stocks. And then just finally, I should point out that these outputs, these analyses are all very preliminary, and there are plenty of ways that we can potentially improve the validity or the accuracy of the data and the robustness of the models. The first of them is to incorporate the efficiency of sampling the different fish species and size classes. So we've not considered that at all at this point, but if we can have good information on that, then we can potentially get more reliable biomass estimates to analyze for the fish. For the environmental data, we've only obtained data so far at the sites where the benthos was sampled. And I think there are good reasons for focusing on the benthos, but the downside to that is that maybe the environmental conditions where the benthos was sampled, but the fish might not be a great predictor of the fish biomass. So another way to do this is to focus on the fish and just sample environmental conditions where the fish were hauled in, or maybe get the average environmental conditions over all cells within the radius that we're using. And then the last thing to note is that alongside all the environmental covariates we've included in the model, these potential confounding factors, we added month because we thought that might be important, but we've not included any other spatial or temporal correlations in the data. So by incorporating that, we can potentially get a more robust model. And the last thing, well, stating the obvious really, is that these results only say something about biomass, which obviously is important from a food web perspective, but it doesn't say anything about other things that we might find important, like species composition or the broader biodiversity of the system. So yeah, I believe Elena in her next talk has something to say about that. So I'll leave it there for now. So yeah, thank you.
Thank you, Derek.